and also in Minnesota. And the wonder about this species, Tyler is the person to go to. And I had also joy of traveling, several times in Alaska with Tyler, and also to different places in Minnesota. And Tyler was always a joy to have in the field, because Tyler has very remarkable, eyes and ears. So if you are with a Tyler, you will constantly hear Tyler saying, "Did you hear the bird sound? And that's such species. Did you hear that sound? And that's such species." So one day, I've been to Minnesota Arboretum many times without seeing owls. But then one day I was finally there with Tyler. But that particular day, Tyler showed me 3 owls. So he has remarkable eyes and ears, and that also helped him to write a wonderful thesis research. So let's hear Tyler what he has to say about jumping owls in Minnesota. Thank you.
Could you get zoom on there so you can, go waiting on all?
Sure. Yeah. And
Well, welcome to my master's thesis defense seminar. So today I'm going to be talking about global warming and the impact of jumping worms in a Minnesota forest. Or not.
Yeah, we can just menu again.
Okay. All right, so today I'm going to be starting my talk with an outline. So first I'll have an introduction about invasive earthworms, and I'll talk about my first chapter research where I looked at how invasive earthworms are spread. And then also my second chapter research where I looked at the impacts of jumping worms. And for that second chapter research, I'll be talking about my research questions, the site descriptions and methods, and then the results, including data about earthworm populations and data about
Interrupt.
so the natural movement of earthworms is really slow, so they would have only moved 50 to 100 kilometers since this last glaciation. So then we have to ask, how did earthworms get here? Originally, they were brought over by European colonizers when they settled in Minnesota.
Recording in progress.
And the recent spread was amplified by globalization.
Yes.
Okay. This is very distracting hearing myself.
Oh.
I'm still hearing myself.
I volume.
Sorry, that's really distracting.
Yeah, I'm gonna turn it off for now. Yeah.
Okay. So the recent spread of invasive earthworms has really been amplified by globalization. So more trade and movement of products has led to more introductions of earthworms. And now I'm going to be discussing a bit about my first chapter where I talked about how earthworms are moved, where I reviewed and compiled research about the human contribution to invasive earthworm transport. So this here is an invasion filter diagram, which goes over how earthworms can successfully become invasive when they're introduced to a new place. So starting on the left here, this is earthworm biodiversity on global scale and local scale. So the global scale includes worms you could get off of Amazon for composting. And then the local scale are worms, say, at the local city works mulch pile, where earthworms are invasive there. So if you grab mulch from that pile and transport it, you can introduce them to new sites. And then moving on, we have the human activities filter. And different human activities really play a role in determining what types of earthworms are moved because certain species are preferentially utilized for certain purposes, including fishing, agriculture, horticulture, and development. So for example, with fishing, people tend to use night crawlers because they have desirable characteristics for that purpose. And then we have the climate and adaptive filter. So these are the conditions during transport and introduction, including the temperature, moisture, and soil properties. So certain types of earthworms won't be able to survive certain types of conditions. So an example of this would be red wigglers in Minnesota, a composting worm. They actually can't survive in forests in Minnesota because it's too cold. So if a species is able to move through these filters, they can become an invading species in a new environment. And here is a deeper look at that human activities filter. So this is a pathways and mechanisms of invasion for earthworms mediated by humans. So the first pathway here is the most intentional end of the scale. This includes the release pathway, which is the intentional introduction as a commodity for release. So things like bioremediation and ecosystem rehabilitation. People are purposely putting worms out in the ecosystem to reap some kind of benefit. And then the escape pathway is the intentional introduction as a commodity, but escapes unintentionally. So this would include something like fishing, composting, gardening, and agriculture. So someone utilizes a worm like a night crawler for fishing, and then when they're done fishing, they dump their excess worms in the woods, unintentionally leading to the invasion of earthworms. And then there's the contaminant pathway. So this is the unintentional introduction with a specific commodity. This includes horticultural materials, mislabeled commodities, as well as construction materials. So an example here is if you buy mulch at the store that happens to be contaminated by invasive earthworms, and then you spread that in your yard, you're unintentionally introducing earthworms with that commodity. And then we have the vector stowaway pathway, which is the unintentional introduction attached to or within a vector. So this includes vehicle traffic, foot traffic, as well as ballast materials. So the example here is if you're hiking in the woods and you step on a patch of mud, you get some earthworm cocoons in your boots, then you go home, clean your boots off, you can move invasive earthworms like that. And then there is the corridor pathway, which is the unintentional introduction via human infrastructure linking previously unconnected regions. So this includes roads and seismic lines, trails, and favorable environments. So someone could create a mulch pile that is watered, and then earthworms are able to preferentially move through that watered area and invade another part of the landscape. And then finally, the most unintentional end of the scale, we have the unintentional introduction of natural dispersal of alien species. So this includes the natural diffusive movement of earthworms, their movement assisted by water, and then their movement through gravity, through things like erosion, and also foresty, which is the movement of an animal species by another animal species. So we could have a robin picking up a worm, flying, and then dropping the worm. So all of these pathways lead to the introduction of invasive earthworms to new environments. So with that, we will move on to some current issues that are happening due to the invasion of earthworms. So invasive earthworms of European origin are ubiquitous in Minnesota due to that long history of invasion from European colonization. So a lot of research has been conducted to understand European earthworm invasion. This work here by Wackett et al. Is actually in Sweden. So we can see that the invasive earthworms are transforming the organic horizon at the top of the soil, and then mixing that with the underlying mineral soil and transforming that into a organic rich mineral horizon, which has very different properties than this spongy organic horizon we used to have on top of the soil. So there is a similar state in Minnesota. Our forests have really been transformed by European earthworms. And these earthworms will continue spreading and continue causing widespread environmental change. And the new issue that is arising is the introduction of invasive earthworms of Asian origin. So these are called jumping worms. And this is what my research is really focused on. Due to their more recent invasion, they have a lot more limited research looking to understand them. And spoiler, they're also going to be able to become widespread in Minnesota. So now we are going to ask, what are jumping worms? And I think there's no better way to really showcase what they are than through some videos. So here we can see I picked up a jumping worm at the Arboretum and it started jumping. So this flailing behavior of the jumping worms is a evolved trait to defend them from predators. And these species are originally from Asia, and they are mainly spread through that contaminant pathway I discussed before, through things like horticultural materials like mulch, compost, and soil. And they are highly invasive and really energetic. So now I've introduced jumping worms, but a little bit more about earthworm ecology and biology. So these are earthworm ecological groups, which are based on a species' feeding and burrowing behaviors. So there are three main groups, including the epigeic group, which involve earthworms that live in the litter layer on top of the soil surface. The endogeic ecological group, which are earthworms that survive in the soil. There's primary soil dwelling, and they make horizontal burrows. And then there's also the amnesic group, which have vertical burrows that are very large. This includes night crawlers, and they actually create these things called middens at the entrance to their burrow. So that's the pile of leaves at the entrance to their burrow, and those are visually apparent in the forest where they're invasive. So European earthworms are from a diverse ecological groups. They're from all three of these groups. They're sexually reproducing, and they are perennial, so they will overwinter in Minnesota, going dormant in the soil over winter. And these come from the Lumbricus, Aporrectodea, and Octolasian genus. So the Lumbricus includes the anesic night crawlers, as well as one other species that is epi-endogeic. So it's actually has an ecological group that is a combination of epigeic and endogeic. And then the Aporrectodea and Octolasian are endogeic European earthworms. So jumping worms are epi-endogeic, so they have that same combination of epigeic and endogeic. They primarily dwell in the litter layer and on the top layer of the soil. They are parthenogenetic, meaning they don't need a partner to reproduce. And they're also annual species, so they senesce in the fall when the temperatures get cold, and then the next generation will hatch in the spring from cocoons. And these come from the Amynthas genus. So here's a jumping worm on the left in the soil that is invaded. So this white band is the clitellum of the earthworm. This means it is mature. And for jumping worms, that clitellum is very white, milky white, and it's very close to the head, which is one way you can identify them. So these pictures really illustrate what jumping worms are doing to the soil. They create this casting layer that's really loose, has the texture of coffee grounds or cat litter, and you can just scoop that up. It's so loose. So this layer was not present before their invasion. So it really has the potential for some large changes to our soils in Minnesota. And now onto my second chapter, research questions. So we're asking these questions as part of the first study of jumping worms in Minnesota, and also the furthest north study of jumping worms in North America. So our first question, how are earthworm populations reshaped following the invasion of jumping worms into previously European invaded forests? What changes to soils do jumping worms generate? Do jumping worms modify soil temperature? And will jumping worms be limited by Minnesota's relatively short growing season? And these questions are all very important to ask due to the limited research of jumping worms in Minnesota and the potential they have for really wide-scale impacts. So our site, our study was conducted at the Minnesota Landscape Arboretum, which is marked in the red border here in central Minnesota. And then our study sites were these 3 forested sites in the center of the Arboretum. And this study was also conducted during a severe drought in Minnesota during 2020 and 2021. So this is what our sites looked like. They were rolling hills, forested sites dominated by sugar maple with some basswood as well. So now looking closer at those study sites, we have 3 transects we established in the forests here, including the Acer transect, the Magnolia transect, and the wood duck transect. So on these maps here, I'm showing our earthworm sampling sites, and the color is dividing them by the population of the earthworms. So we chose these sites because they had 2 end members of different groups of earthworms. So in the white areas, those are the jumping worm dominated areas. And then in these black areas, these are dominated by Lumbricus earthworms, those night crawlers, and then one other species of Lumbricus. So we set these transects up to compare these 2 end members to see the differences in soil properties there. And then these are what these sites look like on the ground. So the jumping worm dominated site is really characterized by that casting layer, that loose soil surface, where you can just scoop it right up. And then the Lumbricus dominated sites are characterized by the middens that the night crawlers make, those piles of leaves at the entrance to the burrow, as well as a dense soil surface. So that image in the bottom right is where I dug some soil and it just comes out in one big chunk rather than a loose layer like the jumping worm sites. And for our methodology, we sampled earthworms by placing this sampling ring on the forest floor and pouring a liquid solution of mustard, which causes the earthworms to emerge out of the ground, and then we can collect them. And then we also dug to 30-centimeter depth and hand sorted the soil to make sure we didn't miss any earthworms. And then we identified those worms to species and measured their biomass. And for soil sampling, we took samples at 3 depth intervals, 0 to 5 centimeters, 5 to 10 centimeters, and 10 to 15 centimeters. And we were measuring the bulk density, the soil organic matter, the pH, and also we took leaf litter samples to measure the leaf litter mass. So these samples were all taken in those 2 end members of each transect so that we could make comparisons. And then we also measured soil temperature using these eye-button temperature loggers. They're these little metal sensors, and we wrapped them up to make sure they didn't get damaged by water. And these were buried at 4 depths. So they were buried above the leaf litter, at the mineral soil surface, at a 5-centimeter depth, and then also at a 15-centimeter depth. And using our temperature data from these loggers, we also calculated growing degree days. But I'll go over growing degree days more when I get to those results. So now we can keep in mind those differences in site characteristics and our methodology and move on to the results that we found. So the first results we're going to look at here are earthworm results in the Acer transect. So this is earthworm population data. On the y-axis, we have ash-free dry mass. This is the biomass of the earthworms at our sites. Grams per meter squared. And then on the x-axis, we have our sampling sites, and those are divided by our seasonal sampling, where we mapped out the earthworm populations throughout our sites. And then the colors are showing the different species of earthworms. So in white, we have the jumping worms, Amynthas, and then in black, we have the Lumbricus earthworms. And then in the shades of gray, we have those endogeeic European earthworms. So we were using this data to answer that question, are jumping worms replacing European earthworms? So the answers to that are yes and no. So at these sites, which had soils indicative of European earthworm Lumbricus dominance with the middens, we found that Amynthas were actually encroaching into those sites and then coexisting or existing there with the Lumbricus. But we also found these areas where we had the endogeic Aporrectodea coexisting with jumping worms in those sites that had the loose casting layer. And moving on here, we have the Magnolia transect. So this is same type of data, same graph setup, just the different transect. And then once again, we have these sites where we found jumping worms in the sites dominated by Lumbricus earthworms. And here we actually did not have any coexistence of Amynthas and the endogeic Aporrectodea. And now the third transect, again, same type of graph. Here we have the Amynthas also in the Lumbricus dominated sites. And then also some coexistence of Amynthas and Aporrectodea. So some takeaways from our earthworm data. This is the most detailed study of population data for jumping worms and European earthworms. And it's the only study of this kind in Minnesota as well. So based on our data, we're finding that jumping worms are replacing the Lumbricus earthworms. So European earthworms were previously ubiquitous throughout these forests. So the night crawlers and the other Lumbricus species were present in all of these forests. And now we have areas where it's just jumping worms and no Lumbricus earthworms. And we also have areas where the jumping worms are moving into those soils dominated by European earthworms, the Lumbricus. We found that jumping worms are not necessarily replacing the Aporrectodea because we did find coexistence at some sites. So some potential reasons for this replacement. First, we have the increased dietary flexibility of jumping worms. Jumping worms are able to consume more sources of organic matter like decaying wood and soil bacteria, and they're able to switch to these sources more rapidly than the Lumbricus earthworms are. And also the soil structural change generated by the jumping worms would destroy the tops of those the night crawler burrows, those middens. They destroy that, and then that can inhibit night crawlers from feeding on leaf litter. And then some soil microbial community shifts may play a role in this, as jumping worms have a different microbiome within them, which influences this greater soil microbiome. And some other research has found that these microbial community shifts can inhibit feeding by Lumbricus earthworms. And at our sites, we believe that jumping worms were introduced through contaminated horticultural materials. So there were mulched trails and garden beds near each of our forested sites. So we think the jumping worms came through those. So now we've gone through the earthworm population data, and we see that there's these two distinct areas of those populations. So now we're going to move on to what changes are we seeing in those two end members that are transects. So first, we're going to look at the bulk density in the soil. So on the y-axis here, we have bulk density, which is grams per cubic centimeter. And then on the x-axis, we have the 3 depth intervals I mentioned earlier. And then these are divided by our 3 transects. So in the white, we have the jumping worm dominated areas, and the gray is the Lumbricus dominated areas. So we found that bulk density was lower at all depth intervals and in each transect in the jumping worm dominated areas. And this difference was significant at some of these sites marked by that asterisk. This is 0.05, it's got cut off on the screen. And this difference was greatest at the 0 to 5 centimeter depth interval, where that casting layer is created. So we are the first study to record bulk density data for jumping worms, and then of course the first study in Minnesota as well to report this kind of change in the areas that were previously dominated by European earthworms. And this change to bulk density is really caused by that soil structural change I've gone over, where we move from that really dense soil that comes out in one big piece, and then we move to that loose layer that can scoop up. And it's important to remember that both of these states are very far from the natural conditions that existed in Minnesota. we can think back to that image of the soil horizons where we're mixing in that organic horizon with the underlying soil, which leads to large bulk density changes. So this change to bulk density really has huge implications for other ecosystem processes, like hydrology. If we're changing the soil from this to this, we're going to have different kind of water infiltration, different kind of overland flow through our system. And we're also going to impact the erosion at the site. We found when we were walking around our sloped forest sites on the jumping worm dominated areas, you can just feel it sliding down slope. So there's a potential for a lot of erosion in this forest now. This can also impact seed germination, which occurs in that top layer of the soil. And then this can affect also plant communities. Certain plants might be able to survive in this, or maybe they do better in this. It remains to be seen. And similarly, this can affect soil micro and macrofauna as well. So, fungi, bacteria, centipedes, spiders, all sorts of fauna. So now we've looked at the bulk density, and we're going to move on to some other impacts of the soil, to soil organic matter. So here we have on the y-axis soil organic matter in percent, and then the same 3 depth intervals divided by each transect. And then in the white, again, we have the jumping worm dominated sites, and the gray, the European earthworm dominated sites, the Lumbricus sites. So we found that soil organic matter was greater in the jumping worm areas at all depth intervals and in each transect. And this difference was greatest in the 0 to 5 centimeter depth interval, and it was also significant at some of the intervals marked by the asters. So both these states are again far from the natural conditions where we used to have that spongy organic matter layer on the surface. And this is caused by the redistribution of that organic layer throughout the soil profile by earthworms. And some potential reasons for this increase we found in the jumping worm dominated areas. There could be an increased incorporation of that organic layer, the leaf litter, in the soil by jumping worms. There could be increased dietary flexibility by the jumping worms, like I mentioned earlier, where they can utilize more sources of organic matter like decaying wood and soil bacteria, and then thus increasing total soil organic matter. Changes to soil microbial communities can mediate changes to organic matter decomposition as well. Macronutrient cycling changes to things like magnesium, calcium, potassium, and phosphorus can also impact the soil organic matter. And we might also be seeing some form of organic matter stabilization, where organic matter could be getting locked up in those aggregates that the jumping worms are creating. And these changes are really going to drive shifts in plant productivity and composition, and then soil microbial communities as well, and also to nutrient cycling. So one other change we found in soils that we measured was the changes to pH at our sites. So here, the same graph set up, just on the y-axis, we have soil pH now. And we found that pH was greater in jumping worm areas at all of our sites and at all depth intervals. And this difference was significant, once again, at some of these sites. And then once again, we're seeing the greatest difference at that 0 to 5 centimeter depth interval, where jumping worms are creating that casting layer. And once again, the pH is another state change from the natural conditions in both of our sites. So this is actually a 2-stage increase, because European earthworms have been found by other research to increase soil pH by translocating base cations from deep in the soil profile to the surface, and then also incorporating that litter layer. And then jumping worms are again, seemingly leading to another increase in pH. And importantly, the increase by European earthworms may make it more likely that jumping worms invade a site, because low pH is a limiting factor for earthworms. So any increases to that could increase earthworm survival. So some potential reasons we're seeing this increase. The loss of organic acids from forest floor litter removal. The litter layer leaches a lot of organic acids into the soil over time. So if we remove that litter layer, we could be removing those organic acids. Soil microbial community changes again could shift the organic matter or the pH. And then macronutrient cycling changes could shift the pH. So the form and abundance of things like calcium, magnesium, and potassium could impact pH. And changes to pH will also drive shifts in plant productivity and composition, as well as shifts to soil microbial communities. So both of these communities have ideal ranges of pH where they can exist. So shifting the pH can then impact how successful they are. And we'd also have shifts in nutrient cycling because certain nutrients are soluble or insoluble or more available at different pH levels. So shifts to pH will affect that too. So now we have looked at all the changes we measured in the mineral soil layer. So now we're going to go above that and look at the litter layer above the soil surface. So here we have the leaf litter dry mass data. So it's leaf litter dry mass on the y-axis in grams per area. And then on the x-axis, we have our different sampling timings. So these were taken seasonally throughout our study. And then it's again divided by 3 transects. And we found that leaf litter was greater in areas with jumping worms. And this difference was greatest in the spring and summer as well. And it was again significant at some sites. So we are the first study to document seasonal litter changes with jumping worms. And also, again, both the states are far from the natural conditions from that redistribution of the organic layer. So some potential reasons we're seeing this higher mass in our jumping worm sites. The annual life cycle I mentioned earlier of jumping worms, where they die in the fall and then come back in the spring for cocoons. They have a really low biomass in the spring and early summer. So that would inhibit their ability to consume lots of organic matter. While the European earthworms can emerge in the spring, fully mature, and just get right to consuming the leaf litter. Again, the increased dietary flexibility of jumping worms, they could be choosing to consume different sources of organic matter rather than just the leaf litter, like the decaying wood or soil bacteria. And other studies have found that leaf litter may play a role in jumping worm life cycles as a microhabitat and refuge from extreme temperatures. So that could lead them to not consume the layer as much. And then also that drought I mentioned earlier, severe drought during our site may have more heavily impacted jumping worms due to their surface dwelling habit, where the European earthworms can really burrow deeply into the soil and go dormant during the drought. And then shifts to leaf litter in these forests is going to drive changes to seed germination. Again, seeds really germinate in that top layer of the soil. So any shifts to that top layer are going to affect seed germination and plant communities. And then microbial communities as well. Fungi really depend on that leaf litter layer. So any shifts to that could increase or decrease fungi. And then we can also see changes to nutrient cycling, as there are a lot of nutrients locked up in this leaf litter layer. So if the decomposition is increased or decreased, you might have more or less nutrients being released. So thinking about all these soil changes, we're now going to move on and look at how these changes are influencing soil temperature at our sites. So here is kind of some big graphs of soil temperature. So these are violin plots showing monthly soil temperature data. And then in the green, we have the jumping worm dominated sites. And then in the red, we have those Lumbricus sites. And the temperature is on the y-axis in degrees Celsius. And then we have month and year on the x-axis. So in each of these monthly violin plots, we can see the monthly average with that dotted line. And then also you can see the maximum and minimums, as well as any the distribution of the temperature data. So here in the growing season at the Acer transect, we are seeing lower monthly average, lower maximum temperature, and then lower temperature variability within those jumping worm dominated sites. So this is pretty significant at some of those sites. And now moving to the Magnolia transect. Okay. We're seeing similar data, but not as pronounced at this site. Generally lower monthly averages at the site as well. And then we are unable to do comparisons at the wood duck transect due to some eye button temperature sensors failed during the study, of course. And now this is the similar data, but now we're at 15 centimeter depth. The data from before was at 5 centimeter depth. I mentioned that. So here in the Acer transect, once again in the growing season, we're seeing cooler soils in our jumping worm dominated sites with lower monthly average, lower maximums, and lower variability. And then again, we couldn't do comparisons at the Magnolia or wood duck transects here because of some more sensor failures. And now moving on to temperature from 2021. So this is the next year of data we took. This is at the 5 centimeter depth divided by the 3 transects here. And then in the green and blue, we have the jumping worm dominated sites. And then in the red and yellow, we have the Lumbricus dominated sites. And here, once again, in the growing season, we're seeing lower monthly average in those jumping worm sites, as well as lower maximums and lower variability as well. And looking at the Magnolia transect, we're seeing those same trends here, a little less pronounced than the Acer transect, but lower monthly average throughout the growing season and lower maximums as well. And then finally, for this depth, the wood duck transect, here the similar trends are seen throughout most of the growing season, not as pronounced as the other sites, but we generally see lower monthly average and lower maximums and lower variability in our jumping worm sites. And now moving on to the 15 centimeter depth for the 2021 data. Here we see the same trends again that we saw in the Acer transect, 5 centimeters with the lower temperatures in the jumping worm sites. And then Magnolia transect, same trends again. And finally, the wood duck transect, we are also seeing those trends throughout most of the growing season. And now using that temperature data, we're also able to look at other measures of soil temperature, including growing degree days. So growing degree days are a measure of heat accumulation that we used in the soil. And then we calculated using that equation on the top right, where we took the daily average temperature and then subtracted a base temperature of 5 degrees Celsius from that. And we used 5 degrees Celsius because that is the point where jumping worms die from cold temperature. That was found in some other studies. And then we summed that up throughout our sampling period. And here we found that in the 3 transects, the growing degree days were lower in the jumping worm dominated sites, showing us again that the soil temperatures are cooler in those sites. And then we also, from the time soils warmed up in the spring, we calculated the point when they first reached 1,000, the growing degree days. And that was found to be the first and second week of August is the point where we reached 1,000 growing degree days. And that point has been found by other research to be the time when jumping worms first mature in the summer. So what this is telling us is that jumping worms are maturing in the first and second week of August. And then there's around 3 months there where they are mature and they're able to lay cocoons, which can then hatch the following spring. So using that 1,000 growing degree days, which equates to around 90 frost-free days, Moore et al. 2018 created this map. And the dark gray areas are showing areas where we have those 90 frost-free days. So these are areas where jumping worms will be able to reach maturity during the growing season. And we can see that in Minnesota, it is all dark gray, indicating that the entire state will be able to be invaded by jumping worms. They won't be limited by soil temperature. So this is barring any local site conditions, like they probably won't invade the bottom of the lake. And then they're going to be able to invade even further north into Canada as well. So some key takeaways from our soil temperature data. We are the first study comparing soil temperature in areas of jumping worms and European earthworms. Really the first study of soil temperature and earthworms. So the first wave invasion of European earthworms would have increased soil temperature by removing that organic horizon. The organic horizon really plays a role in buffering the soil temperature and insulating it. But there has been almost no study on that. And then the second wave invasion by jumping worms leads to a decrease in soil temperature based on our data. And this is caused by the increased litter layer we found, as well as increased soil organic matter, helping to reduce heat conductivity and insulate the soil. And then also the creation of that casting layer. So some other lab work by another study found that the casting layer actually reduces heat conductivity and has a dampening effect on the soil. So the creation of that casting layer is going to insulate the soil as well. And we found that jumping worms are not going to be limited by soil temperature in Minnesota. There is around 3 months in central Minnesota where they can reach maturity and then lay cocoons for the following year. And forests are going to become very heterogeneous with diverging soil temperature regimes. So we have the areas that are dominated by European earthworms, and then the areas dominated by jumping worms. The soil temperatures are going to vary there, and that will, really add some complexity to the landscape. So changes to soil temperature will drive changes to whole ecosystem processes. So temperature is really a mediator of a lot of reactions. So this will change things like micro and macrofauna in the soil, organic matter decomposition, nutrient cycling can all be affected by temperature. And then also seed germination. So if we change the soil temperature, different plants are going to have different temperatures where they're most successful. And then this can also affect forest productivity, as we can affect the roots of trees by changing the soil temperatures. So the conclusions of this research, this is the first study of jumping worms in Minnesota and the furthest north study of jumping worms as well. And we found that jumping worms replace Lumbricus species, but not Aporrectodea species necessarily. And we found that jumping worms are modifying soil properties, including bulk density, soil organic matter, pH, and then also the leaf litter layer. And we also found that jumping worms will not be limited by soil temperature in Minnesota, so they'll be able to reach maturity during our growing season. And it is likely that jumping worm invasion will drive a number of cascading ecosystem impacts, like so some future considerations for this work. As this is the first study of jumping worms in Minnesota, there's very limited research, and this is a pretty short-term study, just in 2 years. So longer-term study is definitely needed to understand how earthworm populations and soil properties will continue evolving, what ecosystem impacts are going to result from these changes, ecosystem impacts like changes to forest productivity, plant community shifts, changes to microbial communities, shifts in hydrology and erosion in the forests, as well as changes to nutrient and carbon cycling. And another important thing is, are there any methods to control the spread of invasive earthworms, be it before or after they're transported and become invasive? There is a study going on right now at the Arboretum that is looking at how to eliminate jumping worms from a forest and reduce their numbers, but results are remaining to be seen for that. So I would like to acknowledge the Minnesota Invasive Terrestrial Plants and Pest Center, MIT PPC, and then also the Institute on the Environment, as the funding for this grant came mostly through the MIT PPC, but also a small grant through the Institute on the Environment. And then also the landscape, U of M Landscape Arboretum, where the site was conducted. And then I'd also like to thank my advisor, Kyungsoo Yoo, for all his help and guidance throughout the process. Would have been pretty impossible without all of his help. And then I'd also like to thank the jumping worm team, including Aaron Buchholz, Shuai Wang, Louis Goodall, and then all the Arboretum staff for their help with sampling and coordination out of the Arboretum. And then I'd like to thank my fellow ULab members, Sarah De Laurentiis and Amisena Sierra Garcia, for their help, and Sarah for her help with all the coding for the temperature data in R. And then also my committee, Lee Freilich, Tracy Twine, Laura Van Riper, and then Kyungsoo again. And also my family and friends for their support and companionship throughout the process. So are there any questions?
Wonderful. Yeah.
Yes, Carl.
Carl, that's a great presentation about jumping worms. I work a lot with the horticulturalists,
Mhm.
and I've given talks on compost before. The more recent ones, they've been asking me, well, should we be even using that compost in our garden? Because of the jumping worm problem.
Right.
Based on what you're showing, and I can see there's really detrimental things in these situations, but what are your thoughts in a gardening situation? It seems like there's some benefits, but
Yeah.
I'd like to hear your.
For jumping worms,
Yeah.
there are some benefits, like you could improve the drainage and the aeration, but again, that loose layer on the soil is difficult for certain types of plants to really root in and germinate in. Like Lee has noticed at some gardens, like hostas, you can just pull them out by the roots because the soil is so loose. So
what should I be telling gardeners that use compost? Did I say forget it? Because
well, one way is spreading compost out like on a driveway, on a tarp, and then covering that with like plastic and letting it bake in the sun. Because once it gets above, I don't remember the temperature, but once it gets above the temperature, the cocoons and the worms are going to be dead, not viable anymore.
Okay, that's a suggestion.
Yeah.
Anything else?
We can also freeze the compost.
Okay. So if you freeze it, you get it, freeze it, then who's going to survive?
Well, at our sites, we found that the soils never got below 0 most of the winter. So if it does get pretty cold, then it should kill the cocoons. Yeah.
The compost heats up to a certain level.
I don't remember the temperature. There's like sources online that have that. Maybe like 105 Celsius. I don't know. Off the top of my head. I don't know off the top of my head. Don't quote me there. Well, not that. Sorry.
No, it would have to be,
Fahrenheit. Fahrenheit.
have-
Any other questions?
Is there any is there any difference, or are there any studies related to if those affect more like pine forest or like temperate forest compared to deciduous? Because most of the study was conducted most of that kind of deciduous forest. So what about like those that they have acid, or they tend to acidify more the soil compared to deciduous forest that they
Right.
modify that? If there is any study that compares this, how much is gonna be affected more like the deciduous forest compared to the like temperate forest or pine forest?
Yeah, so I don't know if Zoom could hear that, but the question is, are jumping worms, are there any studies about their effects in pine forests? I haven't really read any that I can recall. Most of it's been done in deciduous forests. Most of the research has been done on the East Coast, because that's where they've been invasive the longest, and most of it's in deciduous forest. So pine forest could be, if jumping worms can't consume those, pine needle litter, that could be a limiting site condition, and they might not invade those forests, but that kind of remains to be seen.
I will read one question from Zoom and then move on to you. So this is a question from another worm enthusiast and Adrian Wackett. So awesome work, Tyler. It appeared that jumping worm impacts on soil properties were largest at the acer site. Do you think that's driven by higher worm biomass there or some other site characteristics? I'm just curious.
Yeah, I'm not totally sure, but I believe there was higher biomass at those sites. Those were pretty sloped site there. It was more sloped than the other sites. There are also gardens near, those were the ones that were closest to gardens. So they had more leaf litter there. And then they also got water, so that could benefit the jumping worms as well. Since our study was conducted in a drought, the earthworm population data is hard to say, what it would look like in a typical year. So we could have a lot higher biomass there during a typical year.
So besides what was just discussed for, in a gardening setting for heating or freezing compost, is there anything as individuals that we could or should be doing to help decrease the spread of invasive worms? Or is it just something to sort of keep in the back of your mind and just be aware that it exists?
Yeah, being aware that it exists is important. For jumping worms, there's more of a political will to control their spread because they're becoming invasive now. Versus European earthworms, they've been here, as long as any of us have been alive. So people don't think they're invasive, so there's really no political will to control them, and they're also pretty much everywhere already. So at the state level, there are some regulations that are going to be coming through. But individually, I think the ways that people transport worms as an individual would be moving things like mulch and compost to a garden or their yard or landscaping. So really being mindful of that and checking if it is contaminated by earthworms. And then also cleaning your boots off can be important as they can spread by, cocoons becoming attached to your shoes.
I have two more questions from the Zoom, and then I will limit the question session to those two questions, and then the committees will move to the other room to do the secret part of this event. All right. So, how deep into the ground? This is a question from Daniel Baumann, and how deep into the ground will the worms go, and can they avoid the first line?
Yeah, so they are going to be in the top 10 centimeters of the soil, usually in the top 5, which is where that casting layer is developed. And then also they'll be in the leaf litter as well. So they don't go deeper into the soil like the European earthworms do. So as soon as it gets below 5 degrees Celsius in the top of the soil, which happens around November, mid-November, they're going to die off.
Okay, the next question is from Nick Jensen. Since jumping worms are primarily surface dwelling, is their modification of soil conditions to that granular texture limited to upper horizon? Or will their continued presence on a site lead to deeper horizons eventually becoming a similar condition?
Yeah, so some of these changes are going to, leach down, like any changes to the temperature can then leach down further into the soil. And then changes to, nutrient cycling can leach down further into the soil. And then that concern we have about the erosion of the worms. If we have a sloped site where they modified top 5 centimeters and there's a big rainstorm, that all erodes away. Then there's going to be the new soil surface where they have the top 5 centimeters, then they do that erodes away, and we can continue, decimating the slopes on our sites. But longer term studies really needed to understand how this erosion is going to play out.
Response. Right, thank you so much for being here and then and for your attention, and give a big hand to there. So committee members, we are going to move to a different room, which is actually nicer than here. We can
So
do
the ones that on Zoom,
here.
104 degrees for 24 hours.
Where are we moving?
For city.
Fahrenheit.
Yeah.
I thought you said.
Yeah, it's yeah, I think it's helpful.
Yeah, okay.
Yeah,
That
Yeah,
very nice presentation.
thank you.
Really good job. It's very impressive. So what's next?
Well, I'm doing a little more wormwood this summer.
Oh, it's about.
Yeah, I started. This has been, Elon's been tough. And that's the main break.
Yeah.
Yeah.
Oh, and Kyle, and thanks for moving all those around. I really appreciate it. Yeah, really
Sorry
helpful.
about this.
But no, I don't know why I did not see this.
Yeah, I don't even know.
Yeah,
Click on it.
yeah.
Yeah.
But now you're good.
I hardly use this screen, so I'm not.
Maybe.